Welcome to part B of this part series that reviews the guidance for industry published by the FDA in January 2010, M3 R2 non-clinical safety studies for the conduct of human trials and marketing authorization for pharmaceuticals. Mind maps are available for this series and can be obtained by sending a blank email to nonclinical@idaconsultants.com. The first part discussed in detail the non-clinical requirements for a standard pharmaceutical product. This part discusses the exploratory clinical trials programs that are available under the new regulations, generally known as microdosing studies. There are 5 approaches to microdosing studies, and I'll discuss each of them in turn. Approach 1 is a program that allows you to give less than 100 nanograms as a single or divided dose with washouts between dosing to a patient. It's excellent for working out PK with a PET study. And all you need to be able to do this is some basic pharmacology, a bit of in vitro target and receptor profiling, and some primary pharmacodynamics for the dose setting, neither of which need to be done to GLP, and a general toxicology study, which is a single dose in a single species. Now that does need to be done to GLP, and no genotoxicity is required. Then there is approach 2, which enables you to give up to 5 doses of 100 nanograms, again with a significant washout between each dose, and you can use an IV substitution for normal oral dosing to get more value from that PK study. And again, it's excellent for doing PK studies with low activity PET. Again, you need basic pharmacology, in vitro target receptor profiling, and primary pharmacodynamics for dose setting. But you do need a general toxicology study, 7 days repeat dose but only in one species, and genotoxicity is not required. The pharmacology again doesn't need to be on to GLP, but the general toxicology does. So they're very fast track ways of establishing if your product's PK profile is suitable for commercialisation and also suitable for treating the indication you're targeting at. Excellent for risk management. Approach 3 is a single dose escalation study. So you're giving one dose to patients, escalating up from starting at a subtherapeutic dose all the way into therapeutic dosing. It's excellent for PK, not probably best avoided for PET, but you're not looking for maximum tolerated dose. That's not what this is about. It's about PK up to your normal expected dosing point. Pharmacology again is the in vitro targeting and receptor profiling and primary pharmacodynamics for dose setting, but you will also need a core battery of safety pharmacology done to GLP, and this includes cardiovascular, CNS and respiratory toxicity testing, and you'll need a general tox package, single dose studies in 2 species, and you'll also need an AIMS test for the genotox. But again, it's quite a minimal package to be able to give doses up to your expected dose points that can be very powerful. And then there's 2 more approaches which are very similar, approaches 4 and 5, they're bundled together, and this enables you to give a 2-week dosing repeat dose study in man for looking for PK and PD. Again, you're not looking for a maximum tolerated dose at this stage. The difference between 4 and 5 is the starting dose and the finishing dose you go to, and how it's quite complex and you need to look at that in detail, but just bear in mind that there are 2 approaches, approach 4 and 5. The pharmacology for both these approaches is the same as that for approach 3, which is the in vitro targeting receptor profiling, primary pharmacodynamics for dose setting, and the core battery of safety pharmacology, CV, CNS and respiratory toxicity. General toxicity does differ. Approach 4 is 2 weeks of repeat dose in 2 species, a non-rodent and a rodent, quite a comprehensive package. But approach 5 is 2 weeks of repeat dose in rodent, and then all you have to do in the non-rodent is confirm the NOAL, which is the no observed adverse event level. Obviously that NOAL delivers an adverse event, a more detailed study will be required, but if you can confirm that NOAL level, then you're free to go into man on just the basis of a single animal study, which is of huge value. And for both approaches 4 and 5, you will need an Ames test and a chromosomal aberration test for the genotox. The whole benefit of the microdosing is it gives you very early PK, very early pharmacodynamics, early physiology data. You can't have therapeutic intent with your clinical trial design, especially with approaches 1 and 2. If you're entering the therapeutic area with the dose levels at those levels, you may have to reduce that dosing. But again, combined with PET, they can be extremely powerful. They're not applicable to children. As usual, the FDA have confirmed that if you can dose in children, you will require full adult safety data, and you'll also require juvenile animal data as well. But all children trial designs are reviewed on a case-by-case basis. There is no standards beyond that requirement for adult safety data and juvenile animal data. But if you're wanting to do paediatric studies, you'll need to discuss it with the relevant body to get that approved. But as you can see, these are very powerful tools that enable you to fast track into man with very little animal work. So get those mind maps by sending that email off to nonclinical@idaconsultants.com. Also go to the website and have a look there. There's a newsletter you can sign up for, which watches and monitors all the regulations. And there's a whole other bunch of resources that you'll find of value there. If you want to get in touch to discuss any of these aspects, just go to the website, contact details are there, and we always make ourselves available and our expertise available for discussions where people want more data or information. So don't hesitate to get in touch. Thank you for taking the time to look at this presentation, and good luck with all your endeavours. Thank you very much.